So you might have guessed we're interested in, I guess, functional networks in the brain, but not just in kind of a steady state sense, but how these networks interact with each other. And why? Partly because these may facilitate the transfer of information within the brain. There's kind of a long-standing hypothesis in neuroscience that high-frequency activity may organize local information transfer or local information processing, and the lower frequencies organize these larger areas of networks that then can pass information back and forth between different parts of the brain. It's still a theory. We don't know exactly what's going on with all of these networks because there's no really good way to study them right now. You can think about using EEG or MEG. There you're sensitive to the neural activity directly. You've got great temporal resolution, but you're kind of limited to the surface of the brain and you don't have a lot of spatial resolution. fMRI, which I'll talk about more in a minute, is not directly sensitive to neural activity, but it does have this kind of whole brain coverage that we want. You can think about doing things like putting in microelectrodes, which is great, but it's really hard to put in enough microelectrodes to really cover all of the brain if you're interested in these large-scale networks. So, like I said, we're kind of speaking with functional MRI, partly because that's where my background is and that's what I know the most about, and partly because we think it has potential to really give us this whole brain coverage that we need to look at how networks interact. The downside of fMRI is that it's sensitive to changes in oxygenation in the blood, not directly to neural activity. So how do you interpret this? Complicated. So I guess, let me tell you first, before I switch slides here, we usually talk about functional MRI in kind of the traditional sense where you're looking at the response to some sort of task or stimulus. So you take a bunch of images, at some point you turn on a flashing light, take some more images, turn off the flashing light, take some more images, and then look for areas in the brain where the signal changes when you turn on that flashing light. But we're really interested in these spontaneous activity patterns of the networks. And by spontaneous, I just mean that there's no explicit task. I'm not having you count backwards from 100. I'm not telling you to look at a flashing checkerboard. So when we do this in the scanner, we just tell someone to lie in there and rest quietly but not fall asleep. And I should mention that's a lot harder than it sounds like. At least for me. I go to sleep instantly when I'm in the scanner. So we'd like to look at these fluctuations that are linked then to the spontaneous activity. But there are problems when you compare this to looking at task-related activity. The fluctuations that are involved are very small, so on the order of 1% of your MRI signal. They're contaminated with physiological noise, things like respiration and cardiac cycle. Now, those are true for task-related activity as well, although the fluctuations there are a little bit larger, say 2%, maybe 5% if you're doing animal studies. But the main difference between the two is that you can't average when you're looking at the spontaneous activity. There's no time locking. If I want to look at how the brain changes in response to a visual stimulation, I can show you that same flashing checkerboard pattern over and over 10 times and average the response for all 10 times together. I can't do that when I'm looking at spontaneous activity. I don't have any idea of where the time locking is or what I should average to get that. So what's been done with fMRI is to look at something called functional connectivity. So Bharat Biswal came up with this, it's been probably 15 years ago now. And the idea was that if you look at these fluctuations within the gold signal, and you low-pass filter them to get them down into the range where there's less system noise, if there were correlations between the seed region, or the fluctuations in a seed region in one area of the brain, and another region in another area of the brain, that meant that those two parts of the brain were talking to each other. Does that make sense? So he would call this a functional network, because somehow those parts of the brain were coordinating their activities.
So, quick question. So when you say talking to each other, it's just statistically likely to have a correlation. Is that right? Over a long period of time?
Over a long period of time.
Not necessarily a neural connection.
Well, that was the question, and I think that still is the question. So if you say, I think the first study was in motor cortex. They said left motor cortex is strongly correlated with right motor cortex when you take these MR images, and the correlation time we're talking about is for a whole scan, so 5 or 10 minutes. So there was some likelihood that their signals were, more correlated than for the rest of the brain over that period of time. It was interpreted as coordination of the neural activity between those areas. But as it turns out, functional connectivity is a lot more complicated than that. And while there is a neural component to it, I think it's still questionable exactly how much of it is neural. So I'll get to that more in a minute. So for a long time, people weren't even sure that this functional connectivity meant anything, partly because of the contributions of physiological noise and other systems. We know that we're not looking at neural activity directly. And I have to admit that my lab originally started looking at functional connectivity with the whole idea of proving that it did not exist. So we were wrong, and it's good because it's led us to a lot of more interesting approaches now than it would have if we were actually correct. But evidence has been growing. Functional connectivity is linked to intelligence, behavior, task performance. Lots of groups have shown this in humans. People see differences in networks in things like schizophrenia and Alzheimer's disease and epilepsy. You name it, if it is a neurological or psychiatric condition, somebody has done a functional connectivity study and found a difference somewhere. So physicians got really interested in this because it looks like it may have potential to be a biomarker for some of these disorders, even if we don't quite understand what's going on. The complications, again, we mentioned physiological noise. Scanner noise is another problem. Motion is a big problem because if you move your head at all, you get correlation throughout the whole brain. So then it looks like everything's connected to everything. And there's some concern that, local motion is associated with things like cardiac pulsation may influence some of these networks. So if you believe, though, that functional connectivity really reflects some sort of neural coordination, then the question that my lab likes to raise is why not do dynamic analysis? Functional connectivity is measured over 5 or 10 minutes, right? I don't know about you, but the last time my brain was doing the same thing for 5 or 10 minutes, I had to have been asleep or unconscious or something. I'm not even sure it happens then. So we're interested in these shorter time scales, things that may be more relevant to, say, you're lying in the scanner and you're thinking about how much you'd like to go on vacation, and then all of a sudden you remember, oh gosh, I have a math test tomorrow, I need to study that stuff. So we'd like to see those kind of changes, these kind of moment-to-moment changes that may be linked with cognition. And it seems to us that if there's enough neural information and functional connectivity that these minutes-long correlations mean something, there ought to be something in there that tells us about this activity at shorter time scales. So why haven't people looked at this? Signal-to-noise ratio is a big one, and validation is the other issue. So we can map all sorts of dynamics, but how do you know what they mean? We're taking two approaches to this. The one that I'm not going to talk to you a lot about today is to link these dynamics to behavior in human subjects. That's probably the least invasive way to say they have some sort of connection to neural activity. Most of what I'm going to talk about today is linking them to electrical activity in the rodent model, where we can go in and make invasive measurements. So first thing we had to do was actually set up the functional connectivity mapping in the rat, because when we were looking at this, people hadn't really done it much yet. Although a lot of the basis for the functional MRI signal in response to a task was originally examined in rodents. So people studied the physiological basis of bull, links to neural activity. Advantage of the rat, you have very well-controlled subjects. They do exactly what you tell them. You anesthetize them, you fix them in place, they don't move, and they will lay in the scanner for 6 hours if you need them to. Unlike any of my human volunteers who get kind of grumpy after about an hour and a half usually. So the downside to using rats, of course, is that we do use the anesthesia. If you don't use anesthesia, there's a lot of stress, and that introduces its own confounds. So almost all functional imaging studies in rodents use anesthesia, and both the neural activity and kind of the vascular tone are altered by these anesthetics, which changes the signal that we're looking at, the gold signal. And of course, then it's hard to give a rat a cognitive task when it's anesthetized and sleeping in the scanner. So this is the stuff we're going to talk about mostly for this time. This is one slice that we're very interested in the rat. It's a coronal slice. We look at primary somatosensory cortex a lot, partly because a lot of the original functional studies done in rats looked at the response to forepaugh stimulation. So this is a very well-characterized area in the rat brain, and we can find this really easily in MRI. Motor cortex is up here, secondary somatosensory areas here, and caudate putamen here. Now, we're especially interested in this network because it has a lot of examples of connections that let us kind of make inferences about different types of connectivity that are going on. So we know that these cortical areas are mostly connected through the corpus callosum. Caudate putamen is not directly connected through the corpus callosum in the rat, so it's an indirect connection. There are also these sort of feedback loops between S1 and CP that also involve the thalamus and other areas. And then there are some sites where there may be some subcortical input in this network. So it gives us a lot of different things to look at. We're doing our imaging on a 9.4 Tesla scanner in the basement of this building. If you ever want to go see it, I'd be happy to show you. It's about the size of a small car, like one of those little zip cars. So it's a teeny little cute thing. And the animals all go in the center right in here. I've got some parameters here if you're interested. Most relevant is that we usually acquire an image every 500 milliseconds or so for the studies I'm going to show here. And we're using either isoflurane or dexmedetomidine as our anesthetics. So these are some of our first results showing functional connectivity in the rat brain. This is just the power spectral density estimate for the bold signal. And we saw this nice low frequency peak that's similar to what they see in humans. So if you filter down to that peak, and then take a seed region and somatosensory cortex, you get this nice correlation map with somatosensory cortex and maybe a little bit of these medial areas. So that was nice. It confirmed that we could do in rats something like what they see in humans. A little bit later, we got better at this and we were able to find different networks. So you can see here's primary somatosensory cortex again, hippocampus. We get strong correlation in the CP, which is very interesting since they're not directly connected, right? But you'll notice that CP is not correlated with S1, even though we know that they are connected. So there's a lot of complicated stuff going on in here. So one of the things that we're doing to try to answer some of those questions is combining MRI with electrophysiology. It's not easy. It took us about 2 years to really get it set up and going. The problem is that the MRI gradients are rapidly changing magnetic fields. Rapidly changing magnetic fields in electrical recording do not go along really well. So you get these giant, interferences in your electrical recording every time you acquire an image. Meanwhile, the electrodes that you use to acquire that electrical signal distort the magnetic field and mess up your MRI image. So you can see here, these were our first images with the electrodes in place, and you see these giant black holes where the electrodes were. So that was a problem. We were a little bit more confident about the electrical recording because if you look at the pulse sequence for MRI, you have an excitation pulse and then image acquisition. And then there's this time, I have a minimum of 100 milliseconds here, but it's usually actually more like 450 milliseconds for most studies we do, where there is nothing going on in the magnet. We're just waiting for the magnetization to relax back to its original state so we can acquire another image. So we were pretty sure that we could get our local field potentials in here. And so that did work. So this is an actual recording of LFPs. Here we're showing the saturation due to the magnetic switching while we're acquiring the image. There's this kind of ring down effect from saturating the amplifier. We managed to get rid of most of that. And you can do a noise correction by averaging that as a template and then subtracting it out and get most of this back. And then we got decent LFPs in the remainder. So here is one of our improved images. We ended up using glass electrodes to come in at an angle, and that provided less disruption so we could actually get decent MRI signal and electrophysiology. And I've got to point out here that we had a lot of help from Dieter Jaeger in his lab in getting this electrophysiology going, because we'd never touched it before in our lives, and we had no idea what LFPs ought to look like. We thought, maybe that was it. So I'm not going to go into a lot of the neural basis of the functional connectivity today, but I'll just show you a few results we got. This is showing, we looked at the electrical activity under isoflurane, and we varied the dose from about 1.5 to 1.8% ISO, so a little bit of a switch in dose. And we looked at different bands of electrical activity and how they correlated with the bold signal at various time lags. Why time lags? If you've ever looked at standard functional MRI data, there's usually this delay time between when the stimulus starts and when you start to see an increase in the MRI signal. And it has to do with the hemodynamic response to the stimulation. So it takes a while to get this overshoot and oxygenation in that area that we see with MRI. So it turns out that the same thing is true when you're looking at the hemodynamic response, in effect, these spontaneous neural oscillations. So for isoflurane, we found that it didn't really matter what frequency of electrical activity you examined. There was strong correlation between the band-limited power in those frequencies and the MRI signal. So we had very strong broadband correlation. This is due to our notch filter at 60 hertz, just artificial. And if you looked at the peak time, it was usually at around 4 seconds, which is comparable to what people see as the peak time for the hemodynamic response before pulse stimulation under isoflurane. So if you look at this for an individual rat, it looks kind of like this. These are individual coronal slices. The lag time is for a bulb relative to EEG. So at 0, that means we line them up so they were acquired exactly simultaneously. And you see that there's some strong correlation, especially here around 4 seconds. We are, in this case, correlating with the electrical activity from the left electrode and just the broadband power. So even though we're recording from one electrode, you see that we also get this correlation with the other side, which is exactly the sort of network that we see when we look at functional connectivity. So it does imply that this neural activity is underlying the sort of correlation we're getting in these areas. And you get the same thing if you correlate with the other electrode. In this case, the signal is not quite as good, but you can still see that other contralateral area. And these are just showing some examples from all of the different rats at the peak time. So the other thing we did with this data was to look at the correlation between band-limited power in different frequency bands from left and right cortex and the bulb power from left and right cortex. And we plotted one as a version, against the other. So this is the correlation in LFP on this axis, correlation in bulb on this axis. So if you look at gamma, beta, etc., most of these, they're not really strongly correlated. Where we did see a strong correlation was for theta and delta. So it seems that theta and delta band-limited power correlation under isoflurane was most predictive of how correlated the bulb signal was going to be. So maybe a low-frequency origin for this functional connectivity that we see. That was really interesting in the light of the fact that the same data, we found the strongest correlation between the local bulb signal and gamma band-limited power, so the high-frequency band-limited power. So it kind of implies that maybe the frequencies that are mediating this connectivity are different than the ones that are driving that local bulb fluctuation. And that fits again with this long-standing theory about, low frequencies coordinating larger distances.
What's delta frequency range?
Like 1 to 4 hertz. I think for this data we didn't go below 1 hertz. So now that we've got something to validate, I want to come back to, why not dynamic analysis? We're ready, we can do it now, great. But being the impetuous and excitable types that we are, we actually went ahead with it before we even got to the tools we needed to validate it. So while we were developing these methods for doing the simultaneous imaging and recording, we were also looking at what sort of dynamics were present in this MRI signal. So if you think about it, the standard functional connectivity is, this group of 200 or 300 images that span several minutes. And you calculate correlation between 2 time courses over that period and you get one number. Well, why can't you look at that on shorter time scales? And that's kind of the first step that a lot of people took, our lab and a few others. They started breaking that up. So instead of looking for correlation over the whole 6 minutes, they started taking 30-second windows or 1-minute windows and doing kind of the sliding window approach. And what was interesting is that people saw changes in correlation over time. They looked fairly significant. Things would be strongly correlated sometimes, weakly correlated others, and anti-correlated even some of the time. So it sounded very cool. I'll show you some data in a minute. The first people did this in humans and looked at areas within the default mode network. After that, people showed that the same sort of variability was present in anesthetized monkeys. that tells us maybe it's not exactly linked to those short-term cognitive events that we were really hoping to see. But there is still something interesting. And we saw the same thing in anesthetized rats. So that's one type of dynamic, and that'll be the first one I talk about. The other thing that we found is some sort of quasi-periodic pattern that's present in this MRI signal from all the rats. And it's a reproducible pattern in the rat. It starts in lateral cortical areas and propagates towards medial areas. And it's quasi-periodic in that it just seems to occur over and over throughout the entire scan. And it's kind of going on in the background. So the other thing we're trying to do is understand what that is and what it means. We first thought it was a function of, using the anesthesia in the rats, but we immediately found it in unanesthetized humans. So it seems like there's some sort of maybe modulatory or global oscillation that we're picking up with the MRI. But anyway, we'll start with the sliding window correlation. So this is just an example of how people do sliding window correlation. Here are your two time courses. You take this window, you move it along the time course, and you calculate correlation for everything within that window. So we used different sizes of windows. After we calculated the correlation, we segmented it into different states, saying, anything between above 0.4 was very strongly correlated, 0.2 to 0.4 was weakly correlated, stuff between 0.2 and minus 0.2 was not correlated, etc. And that was just to reduce the amount of information so that we could start looking at how much time areas spent in each state. So here's an example. This is from one rat. And this is left S2 to left S1. So primary somatosensory cortex and secondary areas within the same hemisphere. When you look at functional connectivity maps of these two things do not look like they're connected. Okay, so, and you can kind of see that if you average this over these 800 images, it looks like it averaged out to around 0, right? It doesn't look like there's a strong shift towards positive or negative correlation there. So we were very excited because we said, oh great, we're missing all of this interesting information. Sometimes this is going up to very strong positive correlation, sometimes it's very negatively correlated. There are all these transient states happening in this rat that we weren't picking up before. And we thought that was very cool. So these are just different window lengths, and as you expect, you get a lot more variability with shorter window lengths. And we thought, well, maybe that means these shorter window lengths are really picking up on these short-lived states that we're interested in as far as cognition goes. And here's just an example of what I meant by segmentation. So we would divide this up into these 5 states. And mostly we did that so we could do this pattern finding and transition time measurement. So then for each of these segmented time courses, we calculated the percentage of the time course that was spent in each state and the number of transitions between them. So these are hard to see sometimes on the screen, but we divided them up. This is all of the interhemispheric pairs there, S2 to S2, S1 to S1, M1 to M1, CP to CP. These are areas within the same hemisphere, and these are areas across hemispheres, but not homologous areas. And if you look at these interhemispheric pairs, all of them are shifted to have more of this strong positive correlation. So you see they spend a lot of their time in this strongly correlated state, and very little time being anti-correlated. The others are more evenly distributed. So these others are kind of the same as what I was showing back here, that we thought was so exciting, that we were seeing these temporary excursions. Well, then we did a control experiment where we matched up the bold signal from different rats, or different scans even within the same rat. So there was no longer any actual timing information shared. Unfortunately, it looked a whole lot like this. And so when you go to this graph here, if you look at random, the amount of time that randomly matched signals spend in each state looks very similar to that for most pairs of areas. So some of this variability that we were so excited about is just coming from properties of the signal itself. There's a lot of post-processing that goes on in fMRI that may give the signals a lot of autocorrelation. So I think this highlights how important it is to have some sort of external validation or some sort of comparison that really lets you know what's random and what's real. The other thing is that it didn't seem to matter which window length we we used, always, most of the areas looked like random. So we had thought maybe the shorter windows were picking up more interesting events, right? Not so, unfortunately. So we looked at the distribution of the correlation then, because when we saw that, we were worried that the areas that we saw that were shifted towards positive correlation may actually just have kind of a positive correlation shift and then the same noise structure on top of it. But it does look like they're actually skewed towards more positive values. So the histograms don't, the blue here is the random, the red is the real data. And so the real data are much differently shaped than the random data. They don't look like the random data just kind of shifted out. And some of that may be ceiling effect. So we were still a little bit concerned about this. This is just some examples for areas that were not significantly different from random. And you see they look very much like the random data. So going on the assumption that those few interhemispheric areas that we saw that look different for random and the S1-M1 pairings that also looked a little bit different for random or real, we went ahead and we kind of reduced the data even further and set everything to be either positively correlated, negatively correlated, or zero, and started seeing if we could find brain states. So that left us with a certain number of combinations, and we went through all of the rats, all of the time courses, and found the most common states. Well, for this network, the most common state by far was having everything, all of these pairs correlated. So correlation was strong within all of them. We did find others where certain correlations were missing. The S1-M1 pairs were most variable. CP was the most variable of the interhemispheric pairs. And we looked at how this plotted out over the rats. These are different rats up here, different scans. And you can see, the most common states occurred in all the rats, but some of the less common states were driven by a single rat, which is interesting and bears further investigation. So at the time we did this, we didn't actually have any of our simultaneous imaging and recording going to look at this. And we didn't know if it meant anything. I mean, how do you say? It was very difficult. And I think our experiments had actually raised questions about a lot of the approaches people were taking in humans and whether that meant anything either. So we just recently got this paper finished, and it's out under review now. And it shows that if you look at the simultaneous recorded data now, along with the gold data, if you look at correlation between, say, delta and delta from each hemispheric data and theta from left and right hemisphere, etc., you get a same sort of variability over time. So this is correlation in band-limited power from the different frequencies, from low here to high here. So I know this is a very messy graph, but if you follow any single one of these, you'll see a lot of excursions between negative and positive correlation. So same sort of variability we saw with gold. And so then what we can do is we can take that gold sliding window time course, take these LFP sliding window time courses, do that same sort of shift to get the hemodynamic delay, and look for correlation between them. And what we found is that at about 4 seconds again in this isoflurane data, there is very strong correlation between high beta and gamma, and sometimes low beta, sliding window time courses for LFPs and the gold signal. So it looks like at least some of this variability really is due to transient neural synchronization, which we're very excited about now, because that brings us back to maybe we can use this. Limitations are that this is only done for left and right S1 right now. We don't have the data from all the pairs of areas to really look at some of those pairs. I'm especially interested in the pairs that look completely random, because just because something looks random doesn't mean that it is random. And it would be nice to know if there is an underlying neural basis for that too, or if some of this is just, completely properties of the gold signal. So we looked at the correlation between the different bands in the LFP and the gold correlation. And it depends on whether you use global signal regression or not. It's a processing question for fMRI that's still kind of out there. But the higher frequencies, in any rate, are always the ones that are most predictive of this dynamic pattern of the sliding window correlation in gold. Which again is kind of interesting, because when we were talking about this before, we saw that the lowest frequencies were most predictive of the functional connectivity over minutes between different hemispheres. So trying to reconcile these findings, acquired from the same data basically, is still something we're working on. We think maybe anesthesia effects, because before we were looking at it as a function of kind of anesthetic level, might have accounted for some of the changes we saw in the low frequencies. So we also looked at window length dependence. This is just correlation between left and right gamma, left and right high beta, etc. And for all of them, we kind of got a plateau around 50 seconds or so. And if we look at the correlation between bold and LFP, it was a little bit different. This is, again, somewhere we thought that shorter windows might give us more sensitivity. But we didn't really see that. There's more or less a plateau after you get to a window length of, say, 30 or so. Although there are some temporary excursions in some of the lower frequencies that may be interesting. I would ignore everything up here. These are basically windows that are so long. We had only room for, say, 3 or 4 windows in the whole scan, so the error is really high up here. Okay, so that's the sliding window correlation. I want to change gears now and go back to these quasi-periodic patterns that we found. This is really the first thing that we saw in the bold data. And we found it because, we were saying, okay, what's in here? The first thing we did was look at the data. We just normalized everything to unit variance, and we played it as a movie, and we said, wow, there's something going on there. And so after that, we had to figure out, well, how do you characterize something going on there? Your eyes are really good at pulling out patterns, but it's harder to teach a computer to recognize what we see by eye. So we took a simple approach and came up with this kind of auto-regressive pattern finding algorithm. I'll show you in a minute. But let me show you some of the templates that we make with this pattern so you can see what sort of dynamics I'm talking about. This is under isoflurane, and it's usually not as pronounced under isoflurane. For one thing, you'll notice you can only really see it in one hemisphere here. That's not typical, but for isoflurane, it's more common to have that happen. So I'll play it again. What you'll see is a little bit of propagation, kind of from this area towards this area, so a little bit lateral to medial or ventral to dorsal, and an alternation between strong positive signal and strong negative signal. I think you can see it better in the dexmedetomidine data, and it's also bilateral, which is far more typical of the patterns we usually see. So if you watch this bit of positive correlation here, we'll kind of move along the cortex, and then the whole pattern will alternate again. So those were the things that we saw, that we found with these pattern finding algorithms. And we also saw something similar in human subjects that involved default mode and task positive mode. So we were very interested in trying to figure out what was going on with this. This is just the pattern finding algorithm we use. Basically, you select a chunk of images. At first, we were selecting a chunk where we knew the pattern was, that we'd seen it visually. It turns out it doesn't matter. The patterns are actually present in the background the whole time, so even if you don't notice it by eye, it's still going on. So you can pick almost any chunk, you do a correlation with these images, you get areas where that chunk has strong likeness to other areas, average those together, make a new template, and repeat until the template converges. And the template is what I was showing you there, so that's the pattern that's going on, and then you end up with a final plot that kind of shows you when that pattern is strong as a function of time. So we were thinking and thinking about it, and one idea we had was that maybe this pattern was linked to these so-called infraslow LFPs. And what I mean by infraslow is I mean the range below 1 Hertz that people discard a lot due to concerns about drift, problems recording, etc. One of the nice features of these electrical oscillations is that they're comparable in frequency to the bold signal fluctuations that we're looking at. So bold is very slow. The spontaneous oscillations are on the order of 0.1, 0.2 Hertz. So we wanted to look for a direct electrical analog of that. People had been comparing bold to electrical activity for a long time, but usually they were using band-limited power fluctuations for higher frequency activity, which makes sense. But it would be fun to see if there was something that directly linked bold to electrical activity. So we did this. We got a DC amplifier, and we recorded in some rats under isoflurane and dexmedetomidine using the simultaneous imaging, and then looked to see if it was correlated with bold. First, we did kind of a coherence analysis between the bold signal from near the electrode and the electrical signal, and we did get strong coherence in these low frequencies. This is compared to some randomized bootstrap data. So for isoflurane, it's mostly below 0.1 hertz. For this dexmedetomidine group, it's got a larger frequency contribution, which kind of fits with stuff that we've seen before in the functional studies. So we also looked then at how bold compared to the different VLP coherence. And so this is now the band-limited power in traditional bands. And again, for ISO, it's very strong in all bands below 0.1 hertz. For medetomidine, you've got this wider frequency contribution, but it's stronger in the lower frequency bands, and mostly in delta and theta here, this green and blue, although everything contributes at around 0.2 hertz. So here are some of the maps. So this is the correlation between the directly recorded low frequency electrical signal and the bold signal now at different time lags and different anesthetics. So for isoflurane, we get a peak at around 4 seconds if you plot the correlation as a function of time lag. And you see that even though, again, as we're recording from one side, you get very strong correlation with both hemispheres. And when you do dexmedetomidine, you get the similar sort of thing. Interestingly, especially in dexmedetomidine, you can see some pattern of alternation that starts to look like those templates that we made from the MRI signal alone. So that was encouraging to us. That said, well, maybe this really is kind of the neural basis for these patterns that we're seeing in bold. It's kind of an attractive hypothesis because it fits very well again with the idea that very slow electrical oscillations are coordinating these large networks. So these now, instead of looking at the template from the MRI signal, we're looking at correlation between the DC or infraslow activity and the bold signal as a function of time lag between the two. So here is under isoflurane. So you see kind of this high intensity followed by low intensity here in the cortex. And then for dexmedetomidine, you get the same sort of pattern of propagation from lateral to medial areas and the same sort of alternation. So that was encouraging enough that we decided to do a direct comparison. So we lined up our ls-fMRI correlations, and this is as a function of time lag. This is the template that we get for these reproducible quasi-periodic patterns from the MRI data alone. And if you just look at it, they look very similar. And it's kind of remarkable considering that, you know, this is made in a completely different method from what we do here. So if you look then at the correlation between those as you shift them next to each other, you do get a very strong peak of spatial correlation between the two patterns. And both the timing and the spatial extents of the patterns are very similar. So while we can't say at this point that we definitely know that that DC pattern is driving this quasi-periodic bold fluctuation that we're seeing, I think the evidence is very strong. At this point, we need to now go in and do some sort of perturbational study to show that, you know, if we can abolish one or the other goes away. So we have correlation but not causality, I guess is what I'm trying to say at the moment. So, just to give kind of a summary and what we're thinking about here, it looks like these sliding window changes in the bold correlation are most likely linked to high frequency changes in coherence between between band-limited powers from different cortical areas. The quasi-periodic bold fluctuations, on the other hand, seem to be linked to these very slow electrical oscillations. And we're trying to do some work now to see how the two interact with each other. You know, do these very infraslow, do these very slow electrical oscillations actually somehow nest these higher frequencies? And if we looked at the quasi-periodic patterns, would we see high correlation in the sliding window correlation at points where the patterns are particularly strong or at particular phases of the patterns? So are these things nested? And that's something we're trying to work on right now to see if we can figure that out. It's really interesting to us because one of the things that we've seen when we're looking into human studies is that this infraslow EEG pattern has been linked to attention in both normal subjects and in people with ADHD. Similarly, the networks that we see in the humans involving the default mode and the task-positive mode are linked to attentional performance. And one of the things that I did really talk about is we found that the relationship between the two networks in just a short window before a task can predict how fast you react whenever you're watching a dot change cover. So that seems to have some link to attention as well. And so we think maybe what we've got is a way to map this infraslow electrical activity throughout the brain with MRI, and then it might be altered in some of these patient populations. So that's one of the directions we're wanting to take with this. So a lot of limitations to this study. Everything was done under anesthesia, so it's difficult to compare what we're doing in the rats to what you get in humans. One of the things we're trying to do right now is to move to awake rats. We have a special coil that's supposed to hold them with less pain. At least that's what the manufacturer told us. When we actually got it, we have not been impressed with the way it holds the rats or the training protocol that they told us about or anything. So we're redoing all of that. And we're developing a new way to hold the rats in the scanner. And hopefully we will very soon have fMRI data from awake rats that we can examine and see if we see the same sort of variability. We're also planning to take that to do the simultaneous imaging and recording in the awake rats, but that's an extra level of complication, and whether we will successfully achieve that or not is still kind of up in the air at this moment. I'm pretty sure we can get the fMRI though. The other thing we've been doing is looking at human EEG studies, especially with this low frequency EEG measurement. So we've been trying to see if the low frequencies we see in the EEG component in the humans are also linked to the gold signal. And the preliminary data looks like yes. So I'll show you maybe just a slide or two in a minute if we have time. The other thing is we don't really think that the sliding window analysis is the best way to go if you want to map how networks interact over time. So one of the things we're doing is we're trying to move to a wavelet-based analysis where we can cluster areas from the MRI signal based on both their spectral characteristics and their temporal properties, and then see how those interactions change, and really map time-varying networks within the brain. That's something that Jacob's working on, and we're collaborating with an expert in signal processing over at GTRI who's really helping us out a lot with that. So I'll show you just a couple of pictures really quick. I know I'm supposed to end so we can have questions, but I'll give you just a few. So these are examples of wavelet analysis where we've clustered based on the signal, and these line up pretty well with anatomical divisions. The other thing that we note is if you look at around the 0.1 hertz scale, you see a lot of variability in the power, which does suggest again that we can pull out some of this information about changing network analysis. And let's see, this is just a preliminary thing showing that if you look at different networks with the MRI, you do get, this is a time shift EEG versus MRI, you do get correlation with particular electrodes when you look at the very low frequency EEG signal. Again, very preliminary, but this is summed over, I think, 8 subjects, and we are finding electrodes that have some sort of significant correlation with different networks. And vice versa, we can take the signal from an electrode and find significant correlation with the gold signal. This is one that's interesting because it looks again like part of that task-positive network where we see some of the spatiotemporal dynamics in humans. So again, it suggests that maybe these very slow oscillations are really driving some of these large-scale network functions. Okay, so then I wanted to get to the acknowledgments of everybody who did this. This is the lab. You don't have to have a big scary beard to be in the lab, but obviously it helps. I am thinking about growing one myself if I can figure out how to do it. Matt has helped a lot with sliding window analysis and human studies. Jacob's working on the wavelet analysis. Matt has done a whole lot of work on getting the animal functional connectivity up and going. Josh is our human EEG and MRI guru. Windu is our wonderful person who has gotten the simultaneous imaging and recording to really work. It's awesome. Garth is doing a lot of the dynamic analysis, and I had to Photoshop Sadia in, but she just joined our lab, and she's going to be working on helping us find brain states now that we know that some of these dynamic fluctuations really mean something. That's where we're hoping to go next. So with that, I'll stop, and I hope you guys have some questions.